A very good day, my dear undergraduate and postgraduate students. Today, we shall touch upon an important as well as a complex topic, but that which has been repeatedly asked in the examination. The importance of the topic is given in the enclosure, and it is oncogenes. I shall restrict myself to proto-oncogenes and oncogenes in today's short class. Now, what is oncogenesis? Onco means tumor, and genesis is the development of a tumor. Before we go into any topic per se, look at the terms. In this, we should concentrate on proto-oncogene, oncogene, viral oncogene, and cellular oncogene. This is a cycle of cell division. All of us know something about the mitotic phase. There is a prophase, metaphase, anaphase, and telophase. Cells do not continuously keep dividing. They take rest. And then there is a growth and a normal metabolic role that follows. This is the first growth phase. Similarly, the DNA also undergoes a replication. This is the synthetic phase. And finally, there is a preparation for mitosis. This is the second growth phase. This blue line indicates the interface between cell divisions. Let us have this in mind before going further. Proto-oncogenes, a question by itself. The cells divide by mitosis or meiosis. Mitosis happens in somatic cells and meiosis in gametes. So there is a half division, as we call. These are all regulated by several proteins. And the proteins are called cell cycle regulators. Whenever something happens, there has to be a command, and these are coded by genes called proto-oncogenes. So proto-oncogenes are those that are cell cycle regulators. They are normally present and they are normal genes. The production, proliferation, and death of cells are all regulated by proteins synthesized by the proto-oncogenes. However, the proto-oncogenes of some people have got a higher tendency to get converted into oncogenes. And they develop into cancers, causing a neoplasm due to various agents such as radiation, viruses, environmental factors, etc. There are more than 40 proto-oncogenes in humans. By heart. So proto-oncogenes, they are the genes that code for a positive cell cycle. So this is the code. The normal genes, when they undergo a mutation, they become what is called as oncogenes. Now, what is the mechanism? The mutated version of a proto-oncogene is called an oncogene. No other words, please. Mutated version of a proto-oncogene is called an oncogene. And the proto-oncogene encodes for normal proteins, the normal cell division. It inhibits cell differentiation and it halts cell death, the oncogene. Whereas it will prevent the cell differentiation and it will halt the cell death and it causes the cell cycle to continue. Therefore, there is an increased activity and increased chance of malignancy. For the proto-oncogene to become an oncogene, there are certain things which are required. One is mutation. The proto-oncogene itself will have to undergo a mutation so that the normal regulation is not there. There is a loss of regulation or there is a misregulation. Similarly, there can be chromosomal translocations. There are relocations for better expressions, and as a result of which you find there can be a hybrid protein that can be produced and the stem cells can lead to a leukemia. So mutation, chromosomal translocation. These are certain steps which can lead to the activation of the proto-oncogene to oncogene. This is an earlier picture which I had shown under the viruses and neoplasia. Whether it is a virus or it is radiation or chemicals, you find this is a normal cell over here. So this is a normal cell and this is the DNA. You find that there is an entry of the or the activation of the proto-oncogene. And this has to be at a specific spot. It cannot be far away. So when this gets activated, it becomes what is called as an oncogene, an activated oncogene. And ultimately you find that there is a loss of the regulation and it goes out of control, becoming a cancer. So there is a normal cell, change in the nucleus, change in the DNA, and it becomes a cancer cell. The proto-oncogene on its part gets converted into the activated oncogene. Please remember this. These are the references. Now, what are the causes for the change? There are many things such as mutation, gene amplification, and chromosomal translocations. I would like you people to kindly by heart this particular table also. So you find that there is a production of overactive gene products by point mutations. There can be insertions or deletions. So a normal chromosome becomes abnormal. There is an increased transcription or expression by point mutations, insertions or deletions. So it sends a different kind of a signal. And similarly, there can be additional copies of the proto-oncogenes which are produced by gene amplification. There is one defect which is becoming more and more increased in number by gene amplification. The movement of the proto-oncogene into a different chromosomal site. This is important because the proto-oncogene, if it is further away from this site, it might not be effective though it has entered the chromosome or the DNA. Therefore, it goes to a site which is close and this leads to increased expression. Expression is nothing but effectiveness of a property. The fusion of the proto-oncogenes with the other genes can lead to oncogenic activity. Please remember the proto-oncogene by itself is not carcinogenic. It cannot cause cancer. It has to undergo certain changes, and when it undergoes one, and secondly, when it is a correct position, it will lead to the production of activated oncogene, which will lead to neoplasia. This is a wonderful one. What are the mutations and that cause the cell becoming a cancer cell? So look at this one. There is a proto-oncogene. This can undergo various changes. We mentioned point mutation, gene amplification, and translocation or transposition. Three important points. I think this is a wonderful flow diagram which you people can follow. Once there is a translocation or a transposition, you find that there is a normal growth, but it is in excess. Gene amplification. Similarly, the normal growth stimulating protein is in excess. And point mutation, within a control element, you find that normal growth and there is a stimulation in excess, and within the gene, it becomes hyperactive or the degradation is resistant. So any of this can lead to a neoplasm. So I would like you people to kindly understand the resulting product can be a neoplastic cell itself. The proto-oncogene, what are all the various changes, and what is the exact mechanism? This will give you the answer. A few of the proto-oncogenes are given over here, which I would like you people to kindly read through. There may be more, but this is for the understanding. For example, this can be an MCQ, the gene or the growth factor that is involved in a case of Kaposi sarcoma is this. So there can be the change in the growth factor, signal transcription, or regulation, etc., which can lead to a plethora of tumors given in the last column. And this is the protein which is being coded for. Please read this table again. The proto-oncogene, the same thing as I had mentioned earlier, this is the mechanism. So there can be a deletion or a point mutation in a coding sequence, and it leads to a hyperactive protein, and this might lead to a malignancy. Gene amplification, again, a normal protein is being overexpressed, and chromosomal rearrangement, which can lead to a loss of the regulatory sequence, and sometimes a defect can be overexpressed. And also you find that there can be a fusion of proteins which become hyperreactive or active. So this is the mechanism. The next comes the oncogenes. Oncogenes are alleles, and if they undergo a mutation, they act in a dominant or a positive manner, gain of function fashion. So the mutations affect only one allele, one of the either the maternal or the paternal. So this is a normal cell, and you find that there is an abnormal cell. You find that only one chromosome, one region is becoming affected. So this becomes a dominant oncogene. Oncogenes are not special cancer genes, but normal genes that are out of growth control, which get mutated. The genes get mutated, and hence they induce neoplasia. They are out of the regular growth control and repair mechanisms of the cell. The proto-oncogenes are changed into oncogenes by means of mutations, and they are mutated or defective genes as such. There is a change in the DNA sequence or the mutation of the proto-oncogene, which leads to uncontrolled cell division resulting in cancer. They also produce positive regulators keeping the cell alive. So one is proliferation, another one is to be alive. They keep the cell alive until there is a transformation into cancer. Now, what are the differences between proto-oncogenes and oncogenes? So this is an abstract one, and obviously we will have to by heart. Identity. Proto-oncogenes are normal genes, whereas oncogenes are mutated or defective versions of proto-oncogenes. They do not cause cancer, the proto-oncogenes, whereas oncogenes cause cancer. The proto-oncogenes are coded for the normal cell cycle proteins, whereas these are coded for the uncontrolled cell division. The regulation, they are positively regulated as for the normal cells, whereas they are negatively regulated in the sense, once there is a multiplication, it has to stop. But here, it continues proliferation. And second, when things are wrong, it should die, but it is asked to stay alive. Cell cycle regulator proteins. There is an organized function such as cell division and apoptosis. programmed cell death, which is not there in the case of oncogenes. There is increased division, lack of differentiation. Differentiation means resembling the cell of origin, and lack of apoptosis, as a result of which it multiplies, becomes abnormal. DNA sequence is normal, it is changed, or it is defective. Cell division is controlled, whereas it is uncontrolled. This particular one, I think, particularly the students can memorize. The modification here are, it can be genetic, mutation, amplification, gene amplification, or chromosomal translocations. Now, what are the similarities? You find that the oncogenes and proto-oncogenes are genes related to cell division. Both are made up of DNA sequences and both encode for proteins. So this is the reference for this particular. This is derived from the ResearchGate, and this is a list of the oncogenes. The oncogene is given over here, and what is the normal neoplasm that it is associated with, and what is the gene product, and what is the effect as such. For example, in this one, BCL2, it is anti-apoptotic. No apoptotic activity, therefore it continues proliferation. Here it is proliferation, there is a block in the differentiation. Activation of the pro-survival signaling, proliferative. Then proliferation as well as activation of the pro-survival signaling. Activation, pro-survival signaling. And I would like you people to do this. Prosenescent, it allows the cell to become old but not die. So these are the list of oncogenes. I think the students will be knowing this well. You can implement it if you are getting a question on the oncogenes or the proto-oncogenes. To be continued.